then we have to realize that bacteria were here many, many years before us. Somewhere on that timeline, bacteria showed up. I don't know if we have that on there or not, but we're talking about that just before class on how small our timeline is, or our recent past is, compared to the whole thing. So bacteria has been trying to get this stuff established for many, 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 many years. And it's been around trying to do it for a long time. So if you look at the prokaryotes, they were the first inhabitants of Earth. And they still inhabit more of the Earth than what we do. Our plasmids are genes that are carrying the antibiotic resistance. These are resistant plasmids. So they select the bacteria with the genes that are resistant to antibiotics, or to the antibiotics. And this is happening more and more all the time. So people take a little bit of the antibiotics and then they quit taking it. And it doesn't kill the bacterium altogether, but it damages them. But that damage is then transmitted to other means of cause mutation and communications between these different organisms, and it's causing the resistance to come up. Or it's getting to the point now where it's hard to find an antibiotic that will kill some of these pathogens. Trophs. Here's the different kinds of trophs. This will help you in your treasure hunt also. What's a phototroph? Let's see if you know these without using the board. What's a phototroph? Photosynthesis, right? Photosynthesis. Sensitive. They will move towards light. Phototroph. They use sunlight to produce food. Phototroph. What's a chemotroph? They get their energy from chemicals. They ingest chemicals and they get the energy. They produce energy from the chemicals. How about an autotroph? A little louder. Makes its own energy. It requires carbon dioxide. So give me an example of an autotroph. Autotroph. Some of them are very big, some of them are very tiny. How about plants? Plants are autotrophs. How about a heterotroph? Give me an example of, what's a heterotroph? I should say what it is first. Energy from other organisms, that's right. So they can require, hetero means from several different places, right? Or two different, male and female, it could be a heterotrophic relationship. But in this case, we get, produces energy from different sources. So we are heterotrophs. We can get our energy from animals, we can get them from plants, either way, it doesn't matter. So we have photoautotrophs, we have chemoautotrophs, photoheterotrophs, and chemoheterotrophs. Lots of different organisms can get their energy from different ways. And that makes it good because if their energy source runs out, they are still viable and they can still survive. So autotrophs need light and carbon dioxide. Chemotrophs need inorganic chemicals and carbon dioxide. Heterotrophs, these are photoheterotrophs, need light and organic compounds to live on. The rotifers and the coliforms, those are important for those. Right? The chemoheterotrophs, they require some organic compounds like food. You throw some food out on the ground, that's an organic compound. Clostridium is one of them. And the Proteus, some fungi are pretty much good at this. There's lots of fungi in this world. and they're pretty fun guys. Now, some bacteria are pretty cool because they can live with or without oxygen. So we have obligate aerobes. Aerobic means that there is oxygen. If it's anaerobic, there's no oxygen. Remember from your cellular respiration? What process is an anaerobic process in cellular respiration? What are those 3 steps? One of those 3 steps was anaerobic. Those are the 3. Which one is anaerobic? Glycolysis. That's right. So organisms that are obligatory anaerobes have to have oxygen to survive. So obligatory anaerobics, these are poisoned by oxygen. Botulism, gas gangrene, all are these types of processes that take place with obligate anaerobes. They don't need it. Tetanus is another one. You go out in a horse barn, drive a nail into your hand, deep inside it heals over, the bacteria are still in there and they can come out, they can grow without oxygen and they'll kill you. They'll create tetanus bacteria and lock your muscles up and make a mess. Facilitative anaerobes, they can survive with or without oxygen. Pretty cool because you think you got him killed and you can't. Now, Michael Jackson, he slept in an oxygen and a pressure tank to help alleviate some of these problems. Right? If you have organisms that are growing in your body, the theory is if you decrease the oxygen or increase the oxygen, they can't survive. If you take hydrogen peroxide, 35% hydrogen peroxide, and put it on a wart, you will kill that wart because it has too much oxygen. We live, and bacteria live, in a very small equilibrium. If we get above or below that, they are susceptible to death, just like anything else. Prokaryotes are no different. They are bacterium, so we have to look at what they can do. Now, some of them are pretty good because they help us fix nitrogen. They convert the atmospheric nitrogen into ammonia, and they put that into the ground, and the plants can use that as nutrients. Farmers do the same thing. They have these big tanks they pull behind their tractors, put the nitrogen into the ground, and it allows them to grow. Also, lightning can help fix nitrogen into the system. Legumes like soybeans and alfalfa, those are also nitrogen fixers for our soils. And the reason for that is these little microbes are on the roots of those organisms. So, we talked a little bit about this already. In the cyanobacterium anaemia, these are photosynthetic cells and nitrogen fixing. They exchange the metabolic products. These are important. They help each other out with the production. Biofilms are prokaryotes. They are kind of surface coating organisms. You can see this on ponds sometimes, where they go in and they will cover a whole pond in just short periods of time. So, who was it that had the, Josh had the taxonomies the other day about how we classify different organisms in different systems. Well, we do that with bacterium also. Now, bacterium is a little bit different than looking at all the dogs at the Westminster Dog Show, right? You can see the differences in the dogs just by looking at them. Bacterium, we have to look at a microscope. We have to be able to get down to a level that we can see the differences in those microbes. Now, what's cool about bacteria and viruses this way, and especially bacteria, is since they do mutate so much, they can get you sick more than once. Viruses and bacteria, since they mutate, once we build an immune response to it, we should never ever get sick from that bacteria or the virus again. What prevents us from doing so is because we have antibodies built up to that antigen that comes into the body, whether it's a pathogen or dust or whatever. We build an immune response to those substances. But if that substance changes, now everything else changes. Our antibodies don't recognize it, our bodies don't recognize it as something that's been there before. It's like a brand new substance, and we can get sick all over again. That's the difference. Starting to rain. So we have to look at these systematics, how we classify different organisms. Now, as Josh said the other day, we have somewhere in our past, and somewhere in the bacterium's past, we have a universal ancestor. Everything branches from that area, from that person or that organism. So if you see up here, we have our nice little area for eukaryotes. This is us, right? We have gram-positive bacterium down here, cyanobacterium, spirochetes. Okay, those are nasty little things. Chlamydia. Where do we find chlamydia at? Something you need to know if you ever go to a bar and pick up somebody. STIs, right? STDs, chlamydia. Nanobacterium. Nanobacterium, very, very, very tiny bacterium. We found that they are smaller than normal bacterium. Very nasty little things. The book called Calcium Bomb, his theory is that we have nanobacterium in our heart that causes the plaquing in our heart. Not cholesterol, but the nanobacterium are producing cocoons of calcium, and that is stuck inside of our blood vessels in our heart and causes us to have a heart disease, atherosclerosis. Okay, so we have several different kinds, but they all come from a universal ancestor, billions of years ago, literally billions of years ago, probably. So a handful of soil contained can be as 10,000 different prokaryote species. Your hands have thousands of different species of bacteria on them, prokaryotes. The bathroom is filthy with them, right? Tons of it. The table that you're sitting in has thousands of different species and things in our environment. We breathe them in all the time. We touch them, we feel them. They're crawling on our skin, they're everywhere. Make you paranoid at all? No? We'll get there sooner or later. Between parasites and bacterium, we got all sorts of things. Molds, fungus coming at us. We're breathing them in all the time. The beauty is we have an immune system that can take it out. Can detect them, brings it out. So 3 domains, archaea is one of the smallest bacterium. Here's a little chart that shows bacterium, archaea, and eukaryotes. Bacterium, like we talked about, very small bacteria. Then we have archaea, kind of somewhere in between eukaryotes and bacteria. There's a branching point prior to archaea that we have to fall into. Here's some different characteristics of the different organisms. And most of these organisms aren't going to make a lot of sense to you until maybe we study them a little bit deeper, but kind of classifies them into these different areas. Now, we have certain bacteria that are called extremophiles that live in extreme temperatures and pressures and different environments. Right? These are phials. These are specialized little organisms. Many years ago, they found out that there's bacteria in some of these areas that had a lot of volcanic activity, these really hot pools. So they tried to find out what these were. They would scoop them up and bring them back to the laboratory to study them. But when they did, they would die. By the time they got them back to the laboratory, they were dead. They weren't like bacteria that's in normal temperature ranges. They are very heat sensitive. If you get them too cold, they die. Right? They like it really, really hot. And that's where they live. Not all bacterium live in our temperature range. They have different temperatures. Here's a question I usually ask my AMP2 students. If I take you and by yourself, take you to Antarctica, put you into a igloo, just an igloo, you have sterile clothes, sterile food, sterile water, will you get sick? Can you get sick? What do we have to get sick? Has anybody not been sick this winter? Anybody been healthy all winter? Okay, those of you that got sick, what happened to you? Why weren't you like somebody else that didn't get sick? You ever think about that?
Do you have a foreign body introduced into your
foreign body?
To your system or something like that?
Some type of pathogen, right? Something that's going to cause your body some stress. So without that, you can't get sick. So if I put you in Antarctica, everything's sterile, all by yourself, will you get sick?
Maybe. Because wouldn't this stuff mutate eventually? And
What's going to mutate, though?
the bacteria that's already on your body?
It's possible. That could get you sick eventually. Or if you had some bacteria when you went down there, you could get sick. But let's say you went down there and you got sick and you survived it, then could you get sick again?
Eventually.
Eventually? Possible? Probable? Probably not. Because you have to look at, too, the little bacteria that make us sick are also heat very sensitive. If they get too cold, they die. If they get too hot, they die. That's one of the reasons why fever is so effective for us. We increase our body temperature, it kills the bacteria, helps the immune process. But we have extreme halophiles, which live in really salt environments, like the salt mines or salt flats. They have bacteria that are living in there. And then we have thermophiles, very hot environments like at the plumes and down in Hawaii. There's bacteria living in those areas. Around the volcanoes in Yellowstone, there's all kinds of pools with these thermophiles in them. Here's some of them. You can see the different colors as they grow, as they bloom. They're different heat sensitive. Methanogens are ones that live in swamps. These are bacteria, and they produce methane as a waste product. So, but if you look at these, they all send the methane up from below. They are anaerobes. They don't like oxygen, so they're living underneath the bog, underneath the gunk, and the methane travels up the top and gives you that stink. If you stir the bog, you stir the soup, if you will, they die because the oxygen is not conducive for their living. So these little archaea may have been here at the beginning of the earth. This may be what actually started these very extreme organisms. May have been here that could live on other things other than oxygen. You know, at the beginning there wasn't much oxygen on Earth. Lots of carbon dioxide because of the volcanoes spewing that stuff up into the air. Right? So it's kind of fun to watch how the carbon dioxide cycle is in this world. Most of our previous times, in previous years, the volcanoes would spew the carbon dioxide up in the air, it would rain down on the ground, in the form of carbonic acid. Then it goes into the water, and by converting into the water, we get calcium carbonate. We can convert the calcium with our carbonic acid, we get calcium carbonate, and that gives us the layers of our sediment in the bottom of the oceans. So, by us having lots of carbon dioxide and having some nice global warming days in February, we're also helping with that cycle. We're creating more of that happening. So bacteria include a lot of different kinds of prokaryotes. Now, some of them are good, some of them are bad. Not all bacteria, not all prokaryotes are bad. We have several different ones. We have the gram-negative ones. These are photoautotrophs, chemoautotrophs, and heterotrophs. Some are anaerobic, some are aerobic. We have different ones, have different substances or different processes that they do. And they all look different too. Here are some different ones that you can see. They have different shapes, different sizes. But again, if we don't stain them, if we don't give them a color, we can't see them. A lot of them are very tiny and they are clear. So we have alpha probacteria. This is where maybe the mitochondrions came from. With our symbiotic relationships, the alpha probacteria can end up in our cells. Now, we have some simple ones here, the rhizosomes, or ribosomiums. These are the ones that stick on the roots of our nitrogen-fixing legumes, like soybeans, and they convert the nitrogen into usable, fixable ammonia in the dirt. We have the agrobacterium, which are used for making tumors in plants, and we also can use those for genetic engineering because they are plant material, but they're very fast growing, very quickly growing. The beta probacteria, these are soil bacteria. These convert nitrogen into other nitrogen processes, nitrates, and we can use those nitrates for our plants' growth. Some of them we have the gamma protobacterium, and things like Legionnaires disease, Salmonella, cholera. Those are all diseases that at one point in our history were very, very dangerous. Now, Legionella, or Legionnaire's disease, really wasn't a factor until the 70s. They had a big convention, and people got really sick, and they lost a bunch of people because of Legionnaire's disease. It got in the circulatory system, or the circulation system, I should say, of the motel. It distributed to all these people, and they got sick, and some died. Salmonella, where do we find salmonella at? Everywhere, yeah. Turtles have lots of this. Things like lizards, they carry a lot of salmonella on them. And then cholera. This is one that back in the 30s, there was a big cholera outbreak, and it killed thousands and thousands of heads of pigs. It wiped out a farmer in just a couple of days. Nasty stuff. And people got it too. So, Escherichia coli, E. coli as we call it, is in your intestines. All of us have it in our intestines, but it can be very pathogenic too. So if your baby gets E. coli and you get it on your fingers and somehow it gets into your body, you can get sick from it. So, pretty weird stuff. Delta protobacteria. These are slime. Slime secreting myxobacteria. They're pretty cool. They have these little fun fruiting bundles that come off of them, and they produce this slimy mixture, very slippery. Epsilon protobacteria that has the Campylobacteria. It causes blood poisoning, and Helobacteria pylori. These cause stomach ulcers. A lot of people have ulcers. They think they have an ulcer, but it's really a bacterium. This bacterium can live in a very, very acidic environment, and they give you all sorts of problems because of that. So, nasty little critters, and all it takes is an antibiotic to wipe them out. You don't even know you have them after a while. Picture of that. Chlamydia. These bacteria are parasites. They live within animal cells. Chlamydia trachomatis includes the blindness and non-gynecological urethritis in sexual transmission. So this is the most common STI or STD that is out there, chlamydia. And it can be cured because it is a bacteria with antibiotics. So if you get chlamydia, you can go to the doctor and get it fixed. Spirochetes, on the other hand, are not quite so good about getting taken care of anymore. Cyanobacteria and gram-positive bacteria all have different traits, and they can cause different things. Spirochetes are heterotrophs. Treponema pallidum, this is syphilis. What are some symptoms of syphilis? Anybody know? Another one that you should have on your checklist when you go out dating.
Do you get the shakes or something like that?
Yeah, you'll get that, but you'll get a lot more. Syphilis is kind of a nasty one because it goes into the nervous system, and it will eventually make it so that makes you go kind of nutso. Al Capone had syphilis, and they found him fishing in his swimming pool in Florida at some point in his life. By the time you get to tertiary syphilis, it's pretty much your teeth are gone, your brain's rotten, you're toast. Not even human hardly anymore. Borrelia burgdorferi, Lyme disease. These are parasites. Now, Lyme disease, where do we get Lyme disease from? Ticks. What kind of ticks? Deer ticks. What's the difference between a regular tick and a deer tick? Anybody know?
Size.
Which one's smaller?
Deer.
Okay. Ticks are important when you go out in the woods. Not this time of year yet. They're not out yet, but they will be. If it stays warm like this, they'll be out not too long. This is a spirochete. No more difficult to kill, but syphilis can be cured by antibiotics. So we've got two of them so far. Chlamydia and syphilis are curable by antibiotics because they're bacteria. A lot of the others you cannot cure with antibiotics, like things like herpes, congenital warts. Those are all not curable by bacteria, by antibiotics. Had a main break there for a minute. So, photoautotrophs, chloroplasts. They produce oxygen. They need carbon dioxide to work, and they are also put together by a symbiosis relationship. Another picture of plants. Gram-positive bacteria, actinocytomites. These are decomposed soil. Bacillus anthrax. This causes anthrax. Clostridium bacterium. This is botulism. What do you use botulism for? Botulinum. Botox. Very toxic stuff. If they don't kill all the bacterium, you can get Botox. It starts producing the toxins, and you can get sick from it in your body. So they take the botulism or the product from these cells, and they inject it in the skin, and it pretty much paralyzes the muscles, so it gets rid of your wrinkles. You can't smile anymore, but you don't have wrinkles either. Or you can't raise your eyebrows because your muscles aren't able to constrict, or contract, I should say. Staphylococcus and Streptococcus can be pathogenic. Staphylococcus aureus, we find that on our skin. That can also kind of go into this flesh-eating bacterium that we have. And mycoplasm is the smallest type of cells. This is streptomycin. This is mycoplasms. So if we didn't have prokaryotes, our life would be really different. We would not be here. We need these little critters for our biosphere, and we couldn't survive eventually. So if you have a catastrophic event where our bacterium die, we die. We also have decomposers, breaking down corpses, dead vegetation, waste products, all those little things out in the middle of the road that the buzzards can't get to, or squashing it all the time. The bacteria come in and they eat it up. Maggots, those types of things are degenerators, they're decomposers in our body. So we have prokaryotes increase the nitrogen, they help with nitrogen fixing, phosphorus, and potassium for plant growth. That's when you go to the store and you buy some fertilizer, it has different components in it. And the bacterium normally do this. If you can leave a field empty for a few years, it will replenish itself. But most of the fields in Iowa have been farmed so long, that there's not really much happening in the soil. They're pretty much dead. You don't have the bacteria growth that we used to have in the soils. Used to be they would go in, they would let the soil rest for a year or two. And a lot of farmers would put in hay one year and then soybeans one year, and rotate your crops, and then you had a better ground. It made it much more bio-friendly. Now it's corn, bean, just looking at them. I mean, you can't put 660 acres into oats or hay, you just have too much of it. So our farming techniques have changed a lot, and that's not really good for our land or our futures. We kill our land, we kill the bacteria, we kill the prokaryotes in our land, it doesn't produce those nutrients. Without that, we have to keep dumping it in. So, you know, when the ground first was turned over back in the pioneers' time, those people had really lush, beautiful ground, and it grew really well. Plants grew very good because it was all dead, decayed, bacteria, prime stuff. Every year after that, it cuts back more and more to the point now where it's almost sterile. We have to keep putting nutrients in the soil, ammonia, potassium, phosphorus into this with fertilizers to make anything grow at all. Symbiosis is the ecological relationship between two cells. We have the host and the symbiote. Our cells and a mitochondria. That type of thing. Mutualism is both symbiotic organisms benefit. Commensalism, one organism benefits while neither harming nor helping the other one. They're just kind of going along for the ride. Commensalism. Parasitism is where one organism is harmful to the other. Does not kill a host because that doesn't make sense, right? You don't want to kill your host that you're living on. You want to keep them going. It's just like if you don't work, you want your spouse to work because then you don't have to work. And if they quit, they lose their job, then you have to go back to work. And these are all pathogens. Okay, good little stopping point. Let's take about a 5-minute break. I'll finish this chapter, and then we'll go on. Wednesday, then we'll go do a little bit of something else outside. Anything else, we just pile a bunch of leaves up and roll in them or something.